So what we want to look at next is the endomembrane system, and specifically in this recording, the ER. So endomembrane means endo, inside, membrane, system. So a system of membranes inside the cell. Endoplasmic reticulum, inside the cytoplasm, is a network. Reticulum means net-like or network. And when we look at our depiction of the eukaryotic cell, we can see it is quite a network indeed of membranes. So we can list the endomembrane system as the components starting with the nuclear envelope, going to the endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, plasma membrane, and these components are either physically linked together or they're connected by transport or secretion vesicles, which we'll go over as we continue through the system. You can see here the endomembrane system. The arrow then represents the path that a protein will travel through the endomembrane system as it's processed and then packaged and delivered to its ultimate destination, whether that be a vacuole or a lysosome or secreted or part of the plasma membrane. Now, there is some terminology that comes with the ER, and that specifies which type of ER we're talking about. So more than half of the total membranes in the eukaryotic cell are the ER, the endoplasmic reticulum. So that suggests that it's very important. Now, it's continuous with the nuclear envelope, and there are two distinct regions of the ER. There's the smooth ER, which is not going to have ribosomes attached to the surface, and then there's the rough ER, which is going to be studded with ribosomes, hence making it rough. And we can see that in the next slide here. At the very top on the left of the slide, you can see the smooth, those tubular structures, the smooth ER, and then the rough ER just littered with those little red ribosomes, which make it rough. So these two are connected to each other, but they serve different functions biochemically. Now, when we talk about ER or cellular structures, there's another word that you need to be familiar with, and that is the word lumen. So if you look at the strict definition of lumen, it is the inside of a tubular structure. So in cell biology, we'll talk about the lumen of the ER. That's the space inside the ER. The lumen of the nuclear envelope, the lumen of the thylakoid membrane. That's the photosynthetic membrane. But we may also use it on a higher level, that being like the lumen of a blood vessel or the lumen of your small intestine. So again, this idea that the lumen is the space inside is important because sometimes we're going to talk about the biochemistry in the lumen, and sometimes we're going to talk about biochemistry not in the lumen, or to the cytoplasmic side of things. Now, what are the functions of the ER? The smooth ER is going to synthesize lipids, metabolize carbohydrates, detoxify poisons, and store calcium. What does this? Enzymes. Proteins that reside in the smooth ER or reside in the membrane of the smooth ER are going to fulfill these functions because proteins do stuff. And so the smooth ER is a location for them to do their thing. Now, the rough ER, in turn, has bound ribosomes. And those bound ribosomes are there for a reason because they're actually synthesizing a protein called co-translational translocation of a protein into the lumen of the ER as it's being synthesized. And those proteins then are destined to be glycosylated, to become glycoproteins. They then are going to be transported down to the subsequent compartments of the endomembrane system, the Golgi apparatus, and then to secretory vesicles or lysosomes or vacuoles in general. We want to think about the rough ER as a protein processing kind of component of the cell. It's also going to be where we add more membrane to the plasma membrane, for instance, or a membrane factory for the cell.